In the beginning was the Word. This is not a verse in Genesis. This is a verse in John chapter 1. Okay? Because I won't get into the theology behind it. But anyway, in the beginning was the Word, and the Word was with God, and the Word was God. God spoke everything into existence. All things were made by Him, and without Him was not anything made that was made. In Him was life. Outside of Him was the void. Okay? Now, Lindenmayer systems will cover. The Fibonacci series will cover. This is the mathematical. What I'm going to get into right now is the mathematical backbone behind the theory. Unifying equation for the Lindenmayer systems. All right. The rule in Lindenmayer Lindenmayer systems is let A, let B be replaced with A, and let A be replaced with AB. And in this, you're going to see how simple the act of creation really is. It's not complex. We see all this stuff out here, and we think, my God, it must have been so hard to do. I mean, how did He speak every little bitty thing into existence, you know? But we're going to find out here with Lindenmayer systems, it's all based on this principle, and it's actually very simple. And could it happen in an instant? Okay, now, remember this rule. Let B be replaced with A, and let A be replaced with AB. Now, L systems are a mathematical foundation for a theory of biological development proposed by Aristed Lindenmayer in 1968. Simply, it is a mechanism of rewriting character strings, A or B, by replacing parts of the string and is replacing it with A, B, or A. And it's based on a set of production rules set by the designer and then reproduced recursively as if on autopilot. Now remember one of the information theorems we just talked about. All right? This is better observed than described. So that's what this graph shows you. As you see it in line 1, you have B. The rule is, of this progression, this is very, very simple. It can become infinitely more complex than this. But the production rule set by the designer is let B be replaced with A. So from line 1 to line 2, you see A replacing B. And then the other part of that production rule is let A be replaced with AB. So then you see A is replaced with AB. The A is replaced with AB in line 4. The B is replaced with A. So forth and so on. This goes on until the designer sets a stop point. Now look at all the diversity you get on line 6. Just from this very simple production rule that was established by the creator of the system. Better observed than described, which that's it. Now, can anyone see how this applies to the knowledge we have obtained after all these years of experimenting with DNA? This is exactly what I showed you with Mendelian genetics. This is how you get dark hair, blonde hair, red hair, brown eyes, green eyes. It's based on, well, it's a little more complex than this, but it's based on these production rules. A set of production rules was established at the beginning based on acoustics. All right, now, a mechanism for rewriting character strings. DNA is such a character string. But here's the kicker. In order to reproduce recursively, there must be a set of production rules. And in order, as we just discussed in the information theorems, it has to reproduce exactly according to these rules. If it messes up at any time, it destroys the information of the system. This is what mutations does. This is what cancer does. Now, this means boundaries that cannot be crossed or altered. As we saw earlier in the DNA, in the fossil record of musical scales comparison, we saw that it has to go in discrete intervals. This means boundaries that cannot be crossed or altered, the iron rules that we discussed, if the system is going to run on autopilot. These rules are mathematical in nature. Now, is there any biologist's work in history who confirms this? I want to watch how this process works to create a tree, a crude reproduction, growing a tree. Pretty amazing, huh? Charles Darwin's own experiments with pigeons proved this hypothesis, and this is what I was trying to explain earlier, but now here's the math of it. proved his hypothesis false, and proved this mathematical principle to be true. In all his work, all he ever produced was more pigeons. He didn't produce any other kind of bird but pigeons with different beak sizes, and different feet sizes, different colors of feathers, different wingspans, different head sizes, and all that kind of stuff. But more than that, he was independently confirming Mendel's work on peas as the foundation of genetics, which follows this same set of rules as does all biology. You see, all this time, Mendel, by the way, was a monk who believed in creation. He believed in creation and created this whole genetic system that we have today. Now, you see, all this time, we have already been teaching creationism and evolution side by side in the school systems. You know, we have this big contention, oh, we can't teach creation and evolution side by side. That would ruin all science. But we've been teaching it side by side all along. We've been teaching evolution and Mendelian genetics side by side all along. But to not allow creationists to declare this science for what it is, and to move forward with the scientific process, is nothing more than hypocrisy and nothing less than censorship of ideas in science. Now, once you set the rule, the pattern will reproduce itself indefinitely until you, the designer of the system, stop the process. And we see this in DNA replication. And, man, I hope I don't misstate this, but it happens at the end of every RNA replication of a DNA strand. Now, and I believe the code is UGA. So, guys, okay, so you got a DNA strand, you got little segments of DNA, AGTC, right? AGTC, billions long, all right? Each little 3-segment section is called a codon. And this is how the RNA reads to replicate DNA. Alright? So there's a specific RNA, I won't get into that, but there's a specific RNA thing here, it's got a little thing that reads this and it replicates it until it gets down to the end of the strand. Well, the end of the strand, all it's doing is following this until the end of the strand tells it to stop. And then at the stop point, there's this 3-letter code. UGA. I hope I'm not mistaken that code, but I believe that's what's right. I just learned this week in anatomy physiology. Fascinating stuff. But anyway, so this little guy here will go on and he'll read each of these codons. Actually, not the same little guy. He only reads this code. Another one of these guys has to come along and read a different section code. And they're all specific for certain sections of code. But this guy here will go on down and look for another one of these so he can replicate it. Kind of complicated. But anyway, it's following that pattern. Okay? Until we get down to this code. There is not an RNA codon reader, because I don't remember what the name of it is right now, off the top of my head. But it's one of these readers that follows this UGA code. And there will be maybe a dozen of these at the end of each DNA strand that's to be replicated. This is the stop sequence that the creator invented so they would know when to stop the process that we're talking about right there. This is in your genes, and this is how you're created. Amazing. But anyway, all right. Now, this is what my life is like here at Western, because ever since I wrote this thing, every single class I take, I get little bits of information like this, and I mean, it just blows my mind every day. It's amazing how everything I'm learning from physics, biology, chemistry, psychology, it all fits into this paradigm. This is the future. But anyway, I won't toot my own horn on that. Now, let's see, every DNA process uses this method for the purpose of replication. You, the designer, set the code, AGTC, and by understanding the complete set of information within each of those characteristics, because they each turn on different types of characteristics in you. All right, and when you combine them together, they turn on certain characteristics. It's very complicated, but it's an amazing process. All right. It's just like orchestrating music. It's just like creating piano music or an orchestra with trumpets and violins and pianos and trombones and tubas and all this kind of stuff. If you, the designer, can pull all this information together and everything that then you can create this amazing piece of music. And you can create billions and millions and trillions, uncannibal compositions from those sets of instruments because they all contain certain aspects of this. Yeah, I'm rambling. Now, you can synthesize strings governed by a set of rules or laws that you establish by which they will continually reproduce until a new command from outside the system changes something. And that's what that is right there. UGA says, stop replicating this string of DNA. We've completed the process. And then it goes on and does something else. Now, this is exactly the new frontier in science that is being explored right now since the advent of Mendel's work began 150 years ago. Darwin's ideas are in direct contradiction to the principles of mathematics, experimentation, observation, and the scientific method. Now, these are some of the designs that you can produce by that system we just described. This is the biology and terrestrial systems of the planet Earth. Now, they can become much more complicated than this, too. All right. Francis Collins said this: when you have for the first time in front of you this 3.1 billion letter instruction book that conveys all kinds of information and all kinds of mystery about humankind, you can't survey that going through page after page without a sense of awe. I can't help but look at those pages and have a vague sense that this is giving me a glimpse of God's mind. Francis Collins, director of U.S. National Human Genome Research Institute, who discovered the pattern of human DNA. Now, but what I want you to realize is that all of these principles also come down to another mathematical principle.